In this video, we'll discuss about folic acid. Another name of folic acid, vitamin B9. It is water-soluble vitamin and responsible for maturation of RBC. That's why it is known as hematopoietic vitamin. In chemistry, this folic acid, it is composed of Deritin ring connected to para-amino benzoic acid and this structure linked with glutamate. Deritin plus FAFA name is called as steroic acid and this complete structure name is called as folic acid. The name is given Latin word folium. Folium means leaf of green vegetables. So this one deridin plus fafa. This one name is called as teretoic acid. This terroic acid combined with glutamate to form folic acid. So important dietary source of folic acid, green leafy vegetables. That's why the name is given folic acid. Coming to active form or coenzyme form of folic acid, it is tetrahydrofolate, which is active form of vitamin B9. Tetrahydrofolate is the active form. And it is responsible for a number of biochemical reaction. Folic acid with the help of NADPH dependent dihydrofolate reductase. Okay, this folic acid with the help of NADPH dependent dihydrofolate reductase. It is converted to dihydrofolate and this dihydrofolate is converted into tetrahydrofolate. It is again NADPH dependent dihydrofolate reductase reaction. And this tetrahydrofolate is the active form of folic acid or coenzyme form of folic acid, vitamin B9. Dihydrofolate, which is responsible for number of biochemical reaction. Coming to that biochemical reaction, biochemical functions of folic acid, the first important biochemical functions of folic acid, it involved in one carbon metabolism. It involved in one carbon metabolism. It is responsible for transfer of this one carbon component. One carbon component means the organic component which contain transferable carbon. So the organic component contain transferable carbon. They are known as one carbon metabolism. Example of one carbon metabolism: methyl group, methylene group. This methylene group is connected with NH N5N10 of tetrahydrofolate. It form bridge in between N5N10 of tetrahydrofolate. CH2 is the methylene which connected with N5N10. This one connected with N5N10 of tetrahydrofolate. It form bridge in between N5 N10 tetrahydrofolate. Another one is methynyl group. Third one is methynyl group. Next one is formyl group. Then hydroxy methyl group. Then formimino component. Formimino group. These are one carbon compound. These are the example of one carbon component. That means organic component which containing transferable carbon. In folic acid in the form of tetrahydrofolate, which is responsible for transfer of one carbon component. Coming to one carbon metabolism, remember that mnemonic here, YEF. F. This methylene tetrahydrofolate which get converted into methynyl tetrahydrofolate get converted into formyl tetrahydrofolate, YEF. So methylene tetrahydrofolate is converted into methynyl tetrahydrofolate, further converted into formyl tetrahydrofolate. This is the entry point. Now this methylene tetrahydrofolate is the irreversible reaction. It is a irreversible reaction. Again converted into methynyl tetrahydrofolate. So it converted into methyl tetrahydrofolate. Sources of one carbon component are the sources for one carbon component. First one is serine, glycine and choline. Serine, glycine and choline is the sources of one carbon component. Here the sources is histidine. This methynyl tetrahydrofolate sources is histidine. Then formyl tetrahydrofolate sources is formate. Here the irreversible reaction is converted into methyl hydrofolate. This methyl hydrofolate responsible for conversion of homocysteine to methionine. This one methyl tetrahydrofolate is responsible for homocysteine to methionine, and this reaction is required vitamin B2. This vitamin B2 is required for conversion of methyl tetrahydrofolate into this tetrahydrofolate. If there is a deficiency of this vitamin B12, this conversion stop, block the synthesis of methionine from homocysteine, and it get trapped as methyl tetrahydrofolate, because this methyl tetrahydrofolate reaction is the irreversible reaction. It get blocked as methyl tetrahydrofolate, and this is known as functional folate deficiency. So this one name is called as functional folate deficiency, that is known as folate trap. In the deficiency of vitamin B12, it causing folate trap. Now this methylene tetrahydrofolate, it is the important thiamidine synthesis. This one is important for thiamidine synthesis. In this formyl tetrahydrofolate, it is important source for this one is important source for C2 and C8 of purine ring. In this way, through this one carbon pole tetrahydrofolate or folic acid responsible for synthesis of thiamine monophosphate. This one useful for DNA synthesis and important in purine synthesis. That's why the folic acid is important in DNA and RNA synthesis, and also synthesis methionine from one carbon pole. The deficiency of vitamin B12, that is known as folate trap. The homocysteine level elevated, and this leads to cardiovascular manifestation. So here that homocysteine level increases means it leads to cardiovascular manifestation to elevate homocysteine level. So methylene tetahydrofolate irreversible reaction converted into methyl tetahydrofolate, conversion homocysteine to methionine. This reaction required vitamin B12. B12 is required for conversion of methyl tetahydrofolate into tetahydrofolate. Deficiency of vitamin B12 block the synthesis of methionine from homocysteine, so trap methyl tetahydrofolate. So functional folate deficiency, it have that name folate trap. This methylene tetahydrofolate important for thiamine synthesis, DNA synthesis. That formula is for purine ring. So DNA and RNA synthesis we need. Then folate traps. Homocysteine level elevate, it causes cardiovascular manifestation. Now coming to deficiency manifestation or symptoms. Three important deficiency manifestation. First one is megaloblastic anemia. The reason for the megaloblastic anemia in folate deficiency. Okay, in folate deficiency, it's responsible for synthesis of pyrimidine, that is TMP. Okay, it is responsible for synthesis of TMP, thiamine monophosphate, and also responsible for synthesis of C2 and C8 of purine. So this way it is responsible for synthesis of DNA and RNA, nucleic acid synthesis. So this one folic acid essential for nucleic acid synthesis, DNA and RNA. Whatever that megaloblastic anemia is due to deficiency of folic acid, they decreases the synthesis or inhibit synthesis of this thiamine monophosphate, and there is decreased synthesis of purine due to there is decreased synthesis of DNA and RNA due to the inhibited DNA or nucleic acid. Inhibited DNA or nucleic acid synthesis, the cell division stop in S phase. Okay, cell division stop in S phase, and cell growth occur without a cell division, which leads to increase in the cytoplasm of RBC, and there is decreases in size of nucleus. Decreases size of nucleus. So due to two factors, increase in size of cytoplasm, increasing size of cytoplasm, and a decreasing size of nucleus, the RBC appear macrocytic, immature. Okay, immature RBC. So decrease the size of nucleus of RBC appear. There is large cytoplasm in RBC as compared to the nucleus, which is smaller in size due to decrease or inhibit synthesis of nucleic acid. That's why there is megaloblastic anemia in folate deficiency. As there is conversion of histidine, this one conversion of homocysteine to methionine, homocysteine level increases, that is leads to homocysteinemia, and increase homocysteine level causes cardiovascular problem and leads to cardiovascular diseases. Then third one is neural tube defect. Neural tube defect in the form of spina bifida decrease folic acid in pregnancy because increasing demand of folate in the pregnancy. So these are the three deficiency manifestation, that is megaloblastic anemia, homocystinemia, and neural defect due to folic acid deficiency. Next, causes of folate deficiency. First one is increased demand, especially in pregnancy. Second, the drugs interaction. Third one is dietary deficiency. Fourth one is gastrointestinal surgeries. These are the folic acid deficiency causes. Increasing demand, mainly pregnancy period. Drugs interaction, dietary deficiency, and gastrointestinal surgeries. RDA normal individual 200 microgram per day. In pregnancy period, it increases up to 400 microgram per day. In dietary sources, important dietary source, vegetable source, spinach, beans, papaya, orange, etc. This one used for treatment of cancer as well as antibacterial agent. So it is used in antagonist. Two important one antagonist important one is sulfonamide. Sulfonamide. It is antibacterial agent which is structurally similar to FAFA. Via the competitive inhibition, it decreases folic acid synthesis in bacteria and inhibit the growth of bacteria. Decreases folic acid synthesis and inhibit growth of bacteria. Second important is amithopterin. Inhibit enzyme dihydrofolate reductase by the competitive inhibition, because this aminopterin and amithopterin, they are structurally similar to folic acid, gets bind with dihydrofolate reductase and inhibit the action of dihydrofolate reductase, due to which there is decreased synthesis of dihydrofolate, which is required for the division of cancer cell and death cancer cell occur. So this is used in the treatment of cancer, that is aminopterin and aminopterin. These both are example of competitive inhibition. Next one is laboratory diagnosis. First one is the peripheral smear, in which there is macrocytic appearance of RBC, also reticulocytosis. So first one is peripheral smear. In this one there is macrocytic appearance of RBC and reticulocytosis. Second one is histidine load test. When we give histidine, this histidine is converted into FICLU, and this FICLU get excreted in urine, but whenever tetrahydrofolate utilized for this formimino group, this tetrahydrofolate is converted into formamino tetrahydrofolate. Remove this formamino group, does not excreted in urine. The excretion of glutamic acid in urine, that indicate deficiency of folic acid. Third important measurement of folate level, serum and RBC. So this is all about laboratory diagnosis in patient with folic acid deficiency, that megaloblastic anemia.